meantime, because you said, okay, so let's jump from a phase 1, phase 2 trial with biomarker to large phase 3. But as a professor of pharmacology, everybody keeps saying that we need phase 2 for dose determination. Yeah. And that could be also a problem because indeed, in some case, and Thomas said it, you can rely on some target engagement to estimate that this dose should work and that dose should work, but sometimes the target engagement are less obvious, and then you need to rely not just on safety aspect. And so, yes, I know what you can say. Okay, enroll 900 people, follow them for 3 years, and you will have the answer. But we switch from a 10 million trial to a 50, 60 million trial.
Yeah, no, I agree. But I think like in a very short manner, there's no way that you can do very reliable dose selection on a clinical outcome. So I'm thinking then back to the Riluzole trials. So then in the end, this is how they did it. They did a 900 patient study with 2 doses or 3 doses, I believe it was, for 18 months. And then they could determine very correctly the dose.
That could be a very good international recommendation to keep at least 2 doses, even in the phase 3 trial.
Yeah. So I see more and more sponsors are keeping that. So a few of the pharmaceutical companies they just keep 2 doses all the way around. For the entire program. Yeah.
It's a good point. So question, maybe we can go back to the slope war. No.
Yes. So I can start with the slope war.
Yeah.
So I would like to reflect on this proposal of like defining like a responder. So what would happen if the slope is for example always decelerating? So there's like a non-linear trajectory, and because I think the slope of the responder analysis works well if it's like a truly linear decline for a patient, because then you can calculate like a deviation from it. So do you have any?
Yeah, this proposal 2 is related also to proposal 1. So you would include also patients with pre-baseline data. That would something novel also. Kind of trial enrichment with pre-baseline data that gives you an idea what type of ALS we are facing with. So I liked your diagram you showed, that we have got totally different patterns in trajectories in during the course of disease, and that would also ease to make this responder definition. And I think that we haven't done a job that all other communities or most other communities have done to destroy the indication. There is nothing like MS. And there's nothing like breast cancer. And all the others destroyed the indication in terms of trials. And to help sub-cohorts to get trials running. And even the data you showed, extending the observation period virtually, if you have more flail leg, or let's say flail arm syndrome patients in this cohort, I wouldn't be surprised that they slow down, because in the beginning they have rapid progression on the arm and then they slow down. So, and it is no miracle. It is just clinical phenotype. So, and the thing is that we haven't controlled this mix of different diseases yet. And this is my major point I want to make. And this could be healed with your diagram, to put it very simply. But I think we have to get rid of doing trials in ALS in general. Yeah. But we have to do trials in lower motor disease ALS, in terasemtiv. And then we can also do CMAP. It doesn't make sense to submit a new biomarker as CMAP. I know it's a Dutch thing.
No, not necessarily, but
and it doesn't make sense to propose this if you also allow patients with PLS, and things like that.
I still have one question, in fact. For how long do we need to collect data before the trial for one particular patient? Is it possible to do that in clinical practice, in fact? Are we able to follow up each patient long enough to be sure that with pre-trial ALS-FRSR values, we are able to predict correctly its own disease trajectory?
Yeah, because Gaelle nicely shows that the earliest may be the best. And you said, Thomas, longest is the best to predict, but the earliest for drug intervention is the best. So we need to find a good equation on that.
Yeah. Okay, so we are discussing about trials in 2030 anyway. So we are not discussing about now and tomorrow. But we have made some positive experience with the introduction of the ALS app in 2020. And now in Germany, 1,000 ALS patients every month do self-rating. And so I think this is something between 15 and 20% of all patients do already self-rating. And I envision the future in 2030 that we have a data set of patients haven't done self-rating for a long time, and then we can also separate between faster progressing, slower progressing, and then we need different time periods for running in, quote unquote, in the shorter disease duration, of course, shorter running in faster progressing, and vice versa. So I would envision, as we talk about in society about digitization and big data and so on. So I would propose also to include this kind of mentality change also to be a co-producer of data from the patient's perspective.
Yeah, but Thomas, what you have done is just great to have this smartphone assessment, but you didn't answer the answer. You're right. Because we asked for when. I mean, do you think that adding more assessment, for instance, every month, instead of having two points within the year before the entrance in the trial, will improve the prediction? For instance, you have a patient and you have just 2 points of ALS-FRS at one time, 12 months before diagnosis, then it goes to trials. And if we have more data before, we will improve the prediction?
Oh, yeah, okay. So we just finished this kind of non-inferiority study between smartphone and clinic with patients with close to 700 ALS patients on smartphones with at least 3 data points. So that might answer part of the question.
Yeah, so I think in general, the more data points you have, the better you can make the prediction. I see only like one operational issue is that you want to include that patient as fast as possible. So if you have diagnosed the patient, preferably the same day he goes into a trial, because then there's still this belief that earlier intervention...
That's a very pragmatical point.
That was exactly my question. Yeah. But what I understand is that you hope to build in 10 or 5 days on data that you are acquiring now in your cohort. But if we want to base efficacy on the slope for each patient, in fact, for how long do we need to collect for this particular patient data before including the patient in a trial, considering that we wish to be able to include the patient as fast as possible after the diagnosis? So that's where I don't understand how we can manage to do that.
I think we don't know yet exactly how many data points we need in what phenotype with that slope. So, and the time frame, yes. So we simply don't know. But I think this is that we should work on, and we can work on.
Yeah,
yes.
so maybe a good thing is that the slope that you observe during trials not only predicted by had the pre-randomization slope. So other factors like your vital capacity, your age, like where the symptoms started, or maybe like some NFL levels, they can all help you to predict what is the randomization slope. So I think we should look at models that combine all of that information, preferably available at diagnosis, and directly classify them and randomize them. Yeah.
Yeah, that's a very good comment. So we now can do a validation, an internal validation of the rating because of NFL. So we introduced it now in clinical practice. When a patient comes into the clinic, it is presented with slope and FRS and total and SVC and PCF, but also NFL. So, and this shows you if there is a delta, pre-slope delta of 3 in 3 months, but the NFL value is just 30 picogram per ml, it doesn't work. So we know that the patient is wrong in doing the self-rating. So I think we have more data, and this is really different from the Riluzole trial. Now we have more data. We have now remote digital assessment on FRS, and now we have also the biomarker. So and to combine things is very synergistic.
Thomas here. Two questions. One, do you think it's interesting to look at the sequence of progression from one region to the other? Because it's not often a way to describe the heterogeneity of the disease, especially to choose for inclusion criteria. First question. And second, you show a very interesting thing on the cough, assist and the cough ability of the patient, which plays a role in the life expectancy, and also is the diaphragm. Do you think if we choose the life expectancy, the survival as a criteria, we should stratify especially on these two criteria of cough and diaphragm evaluation? Yeah.
So to your first point, I completely agree. So it is so important, the topography and the time and spatial pattern, the spreading. So we call it, I don't know whether you call it the same way, but we call it propagation. To separate it from the progression term. Progression is the speed in general, but propagation is the timely pattern, the spatial pattern. So I completely agree. So that's so different, so important, and not mentioned somewhere. I think so. I haven't read something like that in trial protocols. And the other thing is on
cough.
Yeah.
Cough.
I mean, to survival is a worth criteria to evaluate the efficacy of a drug, but what is the measure of survival? It's from diaphragm and secretions mainly. That's why the patient die. So yeah. I think these criteria are not well described, and it could be inclusion criteria if you look especially on life expectancy.
Yes. So I cannot agree more. So I think there are 4 ways of dying in ALS. And it is only communicated basically one. And that is only hyperventilation. It's only a CSF. It's only SVC. So, but I cannot agree more. There is also the obstructive respiratory deficiency, insufficiency. And this is not communicated. So this is a reason why I proposed also to include dysphagia and hypersalivation as part of the equation, and also it's one story: dysphagia, secrete, including secrete management, and PCF. That's one story basically, or very related story, and it is again not mentioned. So phenotypes are not mentioned, and the second way of dying. That is obstruction is not mentioned. And the third way of dying is malnutrition because of dysphagia. it is in a way mentioned because you exclude PEC, but the fourth form of dying is malnutrition because of metabolic reasons. Luc is not in the room anymore, I think, but he would be the one who supported the most. So, and also this way of dying is not part of any trial design.
A question for the three clinicians. As you know, the ALS population followed in ALS centers is only a proxy of the whole population as compared to registers. And because in most trials, the target engagement is not really proved at the individual level, except for genetic trials. What do you think of the clinical relevance of a selection of a subpopulation only based on the rate of progression? And you think that there is a sense for the ELS community and for the ELS doctors?
We are currently fighting to see who will answer the question. Well, in my opinion, when we only select fast progressors patients, when we manage to do it, because we had all this discussion earlier, it's probably a mix of patients with very different molecular targets. So in the best world, it would be the best to have a target engagement that can be proven for each patient, or to be able to select for trial participation patients that we know that for them the target that the drug is targeting is relevant. But it is maybe it's a dream for now, but if we want to succeed, there is so many molecular heterogeneity in the same clinical phenotype. We need to be able to decipher it and to in the same clinical cluster to be able to find how many biological clusters we have.
Yeah, but for instance, in masitinib trial, they propose to exclude very fast progressors and only to select intermediate and slow progressors. Do you think from a clinical point of view there is a real sense of that type of selection?
From a clinical point of view, I think it's mainly driven by the fact that the very fast patient, you have a lot of complications during the time of the trial. You have PEG placement, you have earlier NIV placement, but I'm not sure it is biologically relevant, but maybe you can...
Yeah, so I mostly from a trial perspective, so I think removing the very fast progressing patients makes sense because usually they cannot complete the trial, and maybe they progress so fast and there's a lot of attrition. So they, if we think about like delayed treatment responses, or I think in that realm, yeah, a patient who drops out after 3 months is probably not going to be very beneficial for the trial, and yeah, it's going to be difficult to measure like if the patient then also benefited clinically. It could be, but maybe the disease progression was already so fast, or yeah.
For masitinib, they were also concerned about the fact that it may be less effective. I don't know if it's relevant from a statistical point of view, but it was behind this.
Yeah, I think that's related to it. Yeah, so I think that if the patient is just treated too short, is too little benefit that you can do.
So except for the genetic ALS, it is very hard to make any conclusions from the progression rate, unless if you have cachexia. So this is very proximal phenotypes, trunk onset phenotypes, and that are said to be more inflammatory, and also more metabolic ALS. So I think these are the phenotypes, very aggressive one. If you want to test a drug that aims at anti-inflammation or as a nutritional trial, as we do also. So if you have a drug or intervention like that, then you could, select also outside centers, patients with cachexia, with rapid weight loss, things like that. So, and I would again also see the internet as an option for democratization of trials. So that's a great term. And but you can actually search for patients being, also outside great cities and more remotely, and ask them, do they have weight loss? Do they have rapid loss? On the ALS app, the patients see already the slope. So they see, oh, I have a slope of 1.5 and I have weight loss. So I may be prone for this trial. Things like that. I think there are many options to do selective recruitment into trials.
I guess my question is about the time of enrolling in a trial. One of the speakers mentioned as soon as possible, but maybe that's not a reasonable way to think about getting a person into the trial. Of course, we want to treat the patients clinically as soon as possible, but the objective of the clinical trial is to be able to make a decision whether a drug is useful or not. And given the heterogeneity in all the phenotypes, and also I think you showed the plateau curves, maybe a criteria is not as soon as possible, but if we're looking at the slope especially, because those plateaus, the slope is 0, of course. So maybe we want people just in the plateau or just after the plateau, but not as soon as possible.
Yeah, I can see why you would consider that. My consideration was mainly that the longer you wait, the more damage is going to be done to the motor neurons, and I think this is the thinking a little bit about like the shorter the diagnostic delay or the disease duration, the more benefit theoretically you could achieve still. But I agree with you that if you include a patient who is just in a plateau phase, he's not going to tell you much on that specific outcome, and that's why I think it's also important to think about other outcomes, not only this clinical progression rate, but maybe like other biological signals. And there's too little information yet, but maybe they show a much more consistent decline, and maybe that patient has a very high NFL level, and if we treat it, you can see it going down. So that could be a consideration. But I can also see your point. Yeah.
Okay.
We had one comment on this, because this is for trial efficacy, but if we consider the patient point of view, it is not a disease where you will get better. So the longer you wait to start a treatment, it is lost for the patient. So I think we need to consider that also.
Yes,
Okay.
I have a naive question about the selection of drugs is based on cellular or animal models, and usually it's is using SOD1 or C9, yes. And so the question is, in phase 2, for example, the patients are also selected based on genetic data, SOD1 or C9 or FUS. This is my question, and whether in the phase 3, as you mentioned, maybe it's enlarged to a population which is with lower percentage of patients with SOD1 mutation or C9. So my question is, either maybe there is a bias in the selection of patients carrying on not the SOD1 or C9 mutation. What do you think?
Yeah. So I think it would only make sense to go for specific mutations if you will.
Not specific mutation, gene.
Of the genes, sorry, the genes, yeah, but SOD1 or the C9. If there's really a rationale for the drug to work only in those genes. If you think that it's more sporadic, I would already start from the outset with a broader population.
No, what
maybe I didn't get your question.
When you look at the clinical trial in SMA, for example, it's linked to one mutation in one gene,
yeah.
and it has been showed using different approach that it's efficient. So my question is, if the phase 2 clinical trial in ALS is focusing on drugs which have been shown to be efficient in models for model of ALS linked to SOD1 mutation or C9, you may expect major difference with respect to the genetic origin of the disease.
Yeah, but it also brings, I think, a challenge to the trial design, because you need to have enough patients then with those genes in your trial population to determine whether that treatment effect only applies to those patients or also to patients outside. We have tried that in the past. So for C9, it's possible still because it's about 10%.
But in this case, you will say, so the phase 3 is negative, so it's no more efficient, while in fact its effect is efficient in a subgroup of patients with SOD1 or C9, and this is important research.
No, I completely agree with you. So if we do a phase 3, I completely agree with you that we need to genotype all of the patients in there and do the analysis later on also on the genetic levels. So
but the point is that the patient in the phase 2 are not just selected on gene. They are not. So, but you are right on the fact that the models are not good. I mean, not good. They are good, but they are models. So, but patient could be also considered as model. Next question, Michael.
Okay, I've got a very.
I would like sorry. I would like to add because I think it's a very important point, and it you talked about harmonizing the design, and you Gaelle, you raised the point of the multi-omics data and the importance of grouping the patients. So I think it's very important to collect genetics data and everything to better understand the reason of the failed trials, and also maybe to check, because we talked about the symptoms of the patients, but the underlying cause, the mechanism, it's very important to address that also.
Yes, it's just to add to what you're saying, the disease is 19% sporadic, so we don't not have the choice except to perform trials in patients who are mainly sporadic patients, except if we test a drug which is supposed to target a specific gene or mutation. So that's the first point. And the second point is that in a perfect world, all patients at diagnosis will be entirely genotyped for the main ALS-related genes. And we couldn't dream that we can also assess their inflammatory response and other disease mechanisms which are relevant. So we could propose the patient to participate in a clinical trial which is relevant for his disease mechanisms. And a pluriel, because most probably there are several disease mechanisms we are downstream the initial cause for most patients, because most patients are sporadic. But this is today it's a dream now.
Okay, here. Okay, so can
I would like to continue on your thought and somehow make it more spicy, to somehow not agree with one of your conclusions. You said trials need to be longer and larger. It depends. In genetic cases, and this is coming to your point, trials need to be shorter and smaller. If we have decent information about the disease mechanisms, so let's say the fusion study going on. So the patients in our center being on the drug unblind themselves. So they are stable. They see it on their app that they are not moving for 20 months. And they see that NFL comes down, flattening. And it is unjustifiable for me that trial designs are so unflexible and not adjustable that you have hundreds of FUS-positive patients not be in the position to get the drug, although we know that it works. It works so nicely, but it contradicts to what I said before. Ionis has economic problems that are public. So they are not in the position to open new trial centers. So the reason is that the recruitment rate is low and so on. In this situation, with a good drug, we know that this is a good drug. I would say we need to make trials shorter with less patients. So this is, I promise to make it spicy.
Maybe we can conclude that it depends.
So we have a
This follows up exactly on what you've just said. Okay, you called about responder modes. Okay, now this is crucial for me because we've got a drug in phase 2 in ALS, which is a generic, but it's also just starting phase 3 in Parkinson's, phase 2 in Lewy body dementia, and it's also used relatively widely in Gaucher's disease, and we correspond with them. In Gaucher's disease, it can actually reverse some cases of neuropathic Gaucher's disease. They don't know why, okay? And I'm terrified that's going to be the same situation with us, whereby in a small number of patients, we can actually get a good effect, but it's obviously going to be wiped out by the statistics. So your responder idea is therefore something which I think is absolutely critical and burning urgent for our trial. So how can we get round that?
Easy.
Ruben has to agree with that.
No, we have
I think we need a definition what is a responder. So this is what you proposed. So I think we need to be more flexible. We have to differentiate trial designs in genetic with high, with decent knowledge about the mechanism, and others not, and so on. And so I think we have to be flexible. Also, in terms of duration and also the outcome measures, including some kind of responder definition.
So we have a key message here that we need to tailor for any new drug, a new design, a new perspective. We cannot apply the same recipe for all the drug coming.
I have it's a naive question, but this in the context of a trial where the drug is obviously efficient because in the participants the clinical scales are getting stable and the neurofilaments are lowering from 60%, just like in the tofersen trial. Can this be solved by an interim analysis? Usually it's planned usually to define that there is no benefit to expect and to stop the study for futility, but maybe it can be used to stop the study before for efficacy.
Exactly. No, I completely agree. So I'm a big fan of interim analysis, and I think we should be doing that, especially when the study is larger or longer. But if there's, in case of like overwhelming efficacy or like the drug is so efficacious that you didn't expect beforehand, and then there's always like the data safety monitoring board that at one point says, okay, now it's enough, and the drug stops the trial.
So we have ways to stop a trial if it's obviously efficient or obviously it does not work.
Yeah, no, but I completely agree with Thomas that depends on like the expected treatment effect. So for genetic therapies, indeed, it's a completely different story. And there we expect large effect sizes, but for the average ALS trial, I don't think it's very likely it will be like all of a sudden like a blockbuster. It's very unlikely. It's going to be more like in the realm of a releasal effect, or maybe like a 25% reduction. And that was my point. In those cases, probably it's better to do a little bit bigger and longer study. Yeah.
So it becomes less spicy.
Yeah.
So we have a question there.
Yes, thank you very much. Listening to you, I understand that ALS is a rare disease, but by stratifying the patient, it's becoming an ultra-rare disease. And my question is, why not using methodology and statistical analysis that are used in ultra-rare diseases, such as in mucopolysaccharidosis? I mean, by that single-case experimental design. So we can test the drug and compare the result of the patient to himself, especially as Thomas Meyer was saying, if we get some preclinical data, several preclinical points before introducing the drug.
Yeah, no, that's an excellent point. I think the main challenge in ALS is that it's so heterogeneous. And for most of the rare diseases, they are also heterogeneous. But it becomes very difficult to compare a patient to their own progression rate. And like an N-of-1 trial, what would be good examples, for example, the number of cramps. So I can first treat a patient with a placebo, and then I see a number of cramps, and then I treat with an active drug, then I see no cramps, and then I put them back on placebo, I see back the cramps. So then it's clear it's efficacious. But in terms of disease modifying and like slowing down a progression rate in like a nonlinear disease, that makes it very difficult. But I think there are other methodologies from rare diseases that we can borrow. So I think the most promising now are to use, for example, data that Thomas are collecting, so like external controls. So can we have like an external or digital twins, they're also sometimes called. So can we have like a prediction of what would have been if we didn't treat that patient, and now we observe it. So those are, I think, promising areas. But so it's a little bit in between.
But shouldn't we check the bad result of the former studies to isolate the different kind of patient and compare it to your database and see if we can stratify better the patient?
Yeah. So we have tried it in the past, but so for example on genetic background, so like in presence of SOD1 or C9. But the problem is that the most trials are about 300 patient big, so a good phase 3 study. And if you then choose many strata, then in the end you end up with 5 patients. and what are we going to say about those? It makes it quite difficult still. Yeah. But it's good for.
And if we use like an N-of-1, like it was suggested, we need also to have a certain stability.
Yeah.
And we observe that ALSFS could be curvilinear or have different, so it could be very difficult then to establish it.
Yeah.
We have questions here and there. Okay.
A naive question. What do you think about the future of the placebo in the ALS trial?
Yeah, fair question. I think for now, and that's mainly because it's so variable, that also prevents us from removing the placebo entirely. So the methods that we're working on now is, for example, that we choose very beneficial randomization ratios, so that you randomize, for example, 1:6 or 1:7, 1:8. And so then you do 1 placebo patients on 8 active patients. So in that case, you miss 7 placebo patients, let's say, and those we try to borrow then from external data sets. And the benefit is of still keeping a small placebo is that we can always check if we do like our statistical analysis, was the result the same if I would have used a placebo? But I don't think we can get rid of it totally yet.
I have a philosophical question. What's the difference between a drug that will improve all the patients by 20% and a drug that will only cure 20% but 100% of their disease?
I mean, this is the situation we are facing now. So in SOD1, I mean, I wouldn't call it a cure, but 80% of all patients are benign anyway, and they stop, and we have several of them with improving symptoms. So that's something what I would put in one camp, and at the same time we have what you said. We are modest enough to be satisfied with the lowering of slope by 10%. So I think we are already in this situation that we live together, that we marry somehow 2 expectation models.
For Ruben, you do not speak of new methodologies such as platform trials. What do you think of this in ALS?
I think it's in the end a very good development because it standardizes the design. So it means that all the drugs that are going into a platform, they're all tested in exactly the same way. So the differences that we see in that platform, they are likely because of different efficacy of the drug. So I like it. The problem is that some of the companies don't like it, and I think we need those companies in ALS to keep finding for a treatment. And the problem, namely, is that you're going to randomize a patient to drug A or drug B from company A or company B, and that allows that I can directly compare the efficacy of drug A to the efficacy of drug B. So suppose that one drug reduces has a 25% reduction and the other one a 30% reduction. Yeah, that might be like a market problem. So I like it, but I think the industry is not there yet totally.
I have a comment on that. So I like also the innovativeness in that. What I don't like it, that if you run one placebo group that shares, 4 other arms, you cannot allow a specific, phenotype on the VROM. So because the placebo is also just generic ALS. So, and this is pushed in the same direction. Placebo trials suggest that you have got one ALS, one protocol, one ALS, which is not true.
Well, it might not be not for all. This multi-arm, multi-stage platform might not be for all trials. In fact, we observe that in Parkinson's disease, there's this initiative in UK. There's another one starting in France here in Parkinson's disease, and another one in Norway. And we can share different type of clinical trials with different platform being connected. And it is only for some component to be tested, as Thomas said. So for instance, in France, we will have more phase 2A, phase 2B trial. It will be more inclusive in UK, and so I think there's also a way. It is not the solution, but it could be a solution for some drugs. And you are right. We can't count just on industrial for that. Yeah. So we need, money from the government to start with, and that's exactly what occurs in Parkinson's disease, thanks to the association, and we are here with ALS LA. So it was with Cure Parkinson, with Parkinson UK, with Michael G. Fox, and the foundation, and with France Parkinson. And so we start with association, with government funds, and then we complement with industry, with one arm, for instance. So we don't let them to choose. We take the lead.
No, I think that's very good. And from a cost perspective, like doing trials in a platform is much cheaper than doing every study alone, because you need to do the whole time the setup and arrange all the contracts.
And it's fun for statisticians.
Yeah, and we got a lot of data.
So, is there any remaining question? Yes? Oh, we have the last question.
Thank you. I have a question. I'm not familiar with clinical story, but is it allowed to combine different drugs for clinical studies?
Yeah, great question. I think that would be the future. So probably for sporadic ALS. Yeah, for sporadic ALS. So before we start about the genetic, but for probably like a drug cocktail. That is, I think, is very important, because there's so many different mechanisms that we also learned in the morning and I think yesterday. So you need multiple drugs to combine that. There's one company out there, and that's specifically interested in doing like a combination kind of therapy. So they're thinking about the platform with 5 different drugs, and they also look at making combinations between different drugs. So it's so you can, for example, add 3 arms or 3 drugs together, or 2 drugs or 1 drug, and are those then differently efficacious? So it's a good way forward.
So we have to finish, because it was a hard work for you. Thank you so much. It was so nice, so interesting. Thank you very much, everyone.